and I'm the Chief Medical Affairs Officer at Dyne Therapeutics. I'm sorry that I can't join you in person, but I hope this video provides you useful information. First, I would like to thank Tali and the entire Little Steps organization for the opportunity to present our data from our Duchenne program. It is always a privilege to speak to the Duchenne community, and I want to thank you for your time and attention. I'd like to remind everyone that I will be making forward-looking statements, so please refer to our website if you would like more information. This slide is really important, and I like to start with the slide every time I give a presentation. At Dyne, it all begins with our mission to deliver life-transforming therapies for people living with serious muscle diseases, with Duchenne being a priority. Our mission is woven deeply into our core values and inspires our employees each and every day. Very briefly, I want to talk about the significant limitations that current exon skipping drugs have in terms of dystrophin restoration and functional improvement. There are three key challenges as to why currently available PMOs have significant limitations. The first is nuclear localization of antisense oligonucleotides, or ASOs, to harness cellular machinery. In other words, this challenge refers to the inability of currently available ASOs like exon skipping PMOs to get to the nucleus of the muscle cell where the genetic basis of the disease is. The second is targeted delivery to muscle fibers. This refers to the inability of other drugs to get deeply and evenly spread into muscle fibers so that it is not a patchwork of antisense oligos getting into the muscle fibers, but rather it's distributed evenly in those muscle fibers and deeply. And the third is broad distribution in the muscles that matter in Duchenne. This last challenge is that other drugs cannot get into all the muscles involved in the disease. In the case of Duchenne, other modalities have trouble getting into the diaphragm, which is a muscle needed for breathing. They cannot get into the cardiac or the heart, and they can't get into other places like smooth muscle and the brain. So Dyne developed the FORCE platform to be the solution to overcoming these challenges, and Dyne 251 is being developed as a next generation exon skipping drug. The platform is expressly designed to efficiently and reliably deliver ASOs to skeletal muscle, cardiac, and smooth muscle and target the genetic basis of disease. And here you can see it consists of three components. The first is the Fab, or the antigen binding fragment, or the antibody fragment, which is the top portion of this Y-shaped picture, which is a full monoclonal antibody. And we take this top portion and we designed it to bind to the transferrin receptor 1, or TFR1. TFR1 is highly expressed across the surface of muscle cells, including the heart, breathing muscles, and skeletal muscles, and other skeletal muscles. So the Fab targets the transferrin receptor 1 and uses it to deliver PMO to muscle while maintaining the natural function of TFR1, which is to transport iron into the cell. On the bottom, you can see a clinically validated linker, which we chose because of its demonstrated safety and efficacy in third-party approved products. And this linker enables precise connection of the Fab to the exon skipping PMO. And finally, we have the exon skipping PMO on the bottom right of the slide, which we rationally selected to target the genetic basis of disease. And this is where we leverage the modularity of the FORCE platform. And what I mean by that is for nuclear targeting diseases such as Duchenne, we choose an ASO payload, and in this case, we chose an exon skipping phosphorodiamidate morpholino oligomer, or a PMO, that targets the specific mutation in the DMD gene. And Dyne 251 is our investigational medicine that uses an exon skipping PMO designed to enable skipping of exon 51 to make shortened functional dystrophin. The FORCE platform is designed with the goal of addressing the current limitations of delivery to muscle tissue that I mentioned. And Dyne 251 is really designed to deliver significant amounts of PMO into cardiac and skeletal muscle, including the diaphragm, and lead to high levels of dystrophin that hopefully leads to meaningful improvement in function. And importantly, it's critical to understand the safety and tolerability profile of Dyne 251. As I mentioned, the mechanism of force is very different from other approaches. We're essentially using the cell's natural biology to deliver our investigational medicine to muscle cells. Now, our Fab binds tightly to TFR1, and TFR1, as I mentioned earlier, is highly expressed across the surface of muscle cells, including the skeletal, cardiac, and smooth muscle. Now, the Fab acts as a kind of key that unlocks the TFR1 receptor and allows our PMO to enter those important muscles to target the genetic basis of Duchenne at the level of the nucleus. So in a way, we use a Trojan horse approach to drive the PMO into the cell using the TFR1 receptor without disrupting its normal function of transporting iron into the cells. So the FORCE platform harnesses this natural mechanism of TFR1 receptor-mediated delivery to transport the PMO across the cell membrane. And this is differentiating because we're not using membrane destabilizing agents to deliver the PMO into the cell or to leave the endosome. And by targeting muscle specifically by using TFR1, we have the potential for a wide therapeutic index, resulting in less frequent dosing and chronic administration. Dyne is well on its way towards achieving the promise of FORCE and delivering for patients. And we've delivered exciting data for our Duchenne program, and I'm pleased to share this with you. Importantly, prior to starting our Dyne 251 program, we proactively obtained expert feedback. And very briefly, before I get into the design, I just want to mention that we engaged key opinion leaders or KOLs from various disciplines, including pediatric and adult neurology, cardiology, physical medicine rehabilitation, pulmonology, and physical therapy. And we gathered this feedback from across the globe. We've also received advice directly from patients and caregivers, as well as global advocacy leaders from around the world, including the Duchenne Community Advisory Board, comprised of advocacy leaders from several countries, including Israel, from around the globe. They provided insights based on their living with and being the voice of the Duchenne community, as well as experience with past and current trials. And this input has been critical in ensuring a patient-centric design, which is really important and has absolutely informed our Deliver study. So before I go into the data, this slide shows you a brief overview of that study design. Importantly, it's a phase 1/2 global trial that we've designed to be potentially registrational. Now, the trial is enrolling ambulant and non-ambulant males with Duchenne who are ages 4 to 16 years and have mutations amenable to exon 51 skipping therapy. And patients who have been previously treated with exon skipping therapy can enroll and deliver after a 12-week washout period. The primary endpoints are safety, tolerability, and change from baseline in dystrophin levels as measured by Western blot. And secondary endpoints include measures of muscle function like NSAA and time function test, as well as exon skipping. We also have wearables to allow collection of data such as stride velocity, which has been accepted by the major European Regulatory Authority, or EMA, as a primary endpoint within Duchenne trials. And for more details on inclusion-exclusion criteria, you can visit our ct.gov page or EU clinical trials page. So I'm going to jump right into some of the data, and this particular slide talks about the safety and tolerability data in our ongoing phase 1/2 clinical trial, as I just mentioned. So at a summary level, we saw the safety profile has been favorable for Dyne 251 based on 54 participants enrolled so far in Deliver with over 675 doses administered to date. The majority of treatment emergent adverse events, or TEAEs, were mild or moderate. It's important to note there have been 3 serious TEAEs potentially related to study drug in 2 participants in the 40 milligram per kilogram study group. Both participants are now home and doing well. Other than the 2 participants with those serious TEAEs in the 40 milligram per kilogram cohort, and that's the PMO dose, and they were dosed every 4 weeks, no other participants have demonstrated persistent related anemia or thrombocytopenia or demonstrated kidney injury. Additionally, no participants have demonstrated clinically meaningful changes in electrolytes, including magnesium. All participants in Deliver are now being dosed at 20 milligrams per kilogram. Again, that's the PMO equivalent dose, including participants dose escalated following the placebo-controlled period from starting doses lower than 20 mg per kg, and participants initiated at 40 mg per kg who are now being dosed at 20 milligrams per kilogram following evaluation of the safety profile at 40 milligrams per kilogram. Now, this slide talks about the dose-dependent delivery of the PMO to muscle. So first, let me orient you to the slide structure, which is the same for the next few slides. The gray bar, which you can just barely see in the placebo group, it represents, as I said, those placebo-treated patients. The blue bar are patients dosed at 5 milligrams per kilogram, again, the PMO equivalent dose. The green bar is 10 milligrams per kilogram, and the dark blue bar are 20 milligrams per kilogram. And Dyne 251 is given every 4 weeks with respect to the data I'm showing here. And you can see the change from baseline to 6 months. This slide shows muscle concentration, and you can see very clearly that Dyne 251 drove dose-dependent increase in PMO muscle concentration. This confirms that the drug is getting into the muscles and more gets in with higher doses. We also saw a dose-dependent increase in exon skipping. Here you can see that the 10 milligram per kilogram cohort of Dyne 251 resulted in nearly 2% exon skipping and approximately 3.3% at 20 milligrams per kilogram after 6 months of treatment. So we were very pleased with this level of exon skipping. Now we're particularly enthusiastic about the dystrophin data as measured by Western blot. At the 10 milligram per kilogram dose delivered every 4 weeks, Dyne 251 reached 3.2% normal dystrophin, a change of nearly 3% from baseline to 6 months. And at the 20 milligram per kilogram dose, we see a dystrophin level of 3.7% at 6 months, which is a change of 3.15 from baseline. Now, this dystrophin analysis is unadjusted for muscle content, and we believe this is an appropriate evaluation of dystrophin. And we believe these data are quite striking. We do know that others in the field have adjusted for muscle content taken from the muscle biopsies, so we've done that analysis as well. Here you can clearly see how we have calculated this. MHC dystrophin divided by the percent muscle content. We've been very transparent in how we've come to this conclusion. At 10 milligrams per kilogram, Dyne 251 achieved an approximately 7.6% dystrophin level at 6 months, and this is a change of nearly 7% from baseline. And at 20 milligrams per kilogram, Dyne 251 resulted in about 8.7% adjusted dystrophin and about a 7.4% change from baseline to 6 months. We believe this is the highest level of dystrophin reported for an exon skipping therapy for exon 51 skipping. What's really important and what's really exciting is that we see that this dystrophin production led to encouraging trends across multiple functional endpoints at 6 months and even out to 12 months in the 10 milligram per kilogram cohort. So here you can see that we have the placebo in the gray line, the 10 milligrams per kilogram dosed every 4 weeks in the green, and the 20 milligrams per kilogram dosed every 4 weeks in the dark blue. And what we have here are typical endpoints such as the NSAA, time to rise from floor, and 10-meter walk run, but also a wearable. And here we have stride velocity 95th centile, and I'll talk more about that in a moment. So we see, again, encouraging trends across multiple functional endpoints in multiple cohorts from 6 months to 12 months. We see stabilization or improvement with 10 milligrams per kilogram of Dyne 251, while participants receiving placebo decline over the same period of time when you look at NSAA, time to rise from floor, and timed 10-meter walk-run test, as well as the stride velocity 95th centile. And at 20 milligrams per kilogram of Dyne 251 dosed every 4 weeks, we see functional benefit as early as 6 months. And we see this functional benefit in all of these time function tests, the NSAA, and importantly, the stride velocity 95th centile. So it's really encouraging to see stabilization or improvement at 6 months and the stabilization all the way out to 12 months in the 10 milligram per kilogram cohort. We'll continue to evaluate functional impact at longer time points. And while we recognize that these are relatively small participant numbers, we're really encouraged by this data. I just want to take a moment and talk a little bit more about the stride velocity 95th centile. It's really important to note that it's qualified as a digital primary endpoint by the European Medicines Agency in studies on boys who have Duchenne and that are greater than 4 years of age, greater than or equal to 4 years of age. As I mentioned, it's a digital objective endpoint of ambulatory performance in patients' normal daily environment. So this is a real-world data endpoint. Importantly, it's correlated with the traditional clinical outcomes, such as 6-minute walk test, NSAA, and forced stair climb. And also, it has a defined minimally clinically important difference of 0.1 per second. And what I showed on the previous slide is that we were able to show on the stride velocity an improvement much greater than the minimally clinically important difference in both our 10 milligram and our 20 milligram dose cohorts. So in summary, what we saw is that these delivered data reinforce the opportunity to be able to have meaningful changes in biomarkers as well as function. We saw that with respect to dystrophin, as measured by Western blot, patients treated with the 20 milligram per kilogram PMO equivalent dose every 4 weeks of Dyne 251 had a mean absolute dystrophin level of 3.7% of normal at 6 months. And when we adjusted for muscle content, the Dyne 251 treated group reached about 8.72 mean absolute dystrophin. In terms of function, we saw improvements observed in multiple functional endpoints in the 20 milligram per kilogram cohort, dosed every 4 weeks at 6 months. And this included assessments such as the NSAA, the time function test, and the stride velocity 95th centile. We also saw continued benefit in those endpoints in the 10 milligram per kilogram cohort from 6 months all the way out to 12 months. We saw meaningful improvement in the stride velocity 95th centile in both the 10 and 20 mg per kg study groups following dosing every 4 weeks. And again, SV95C is a digital, real-world objective outcomes measure of ambulatory performance in patients' normal daily environment, and it's approved as a primary endpoint for Duchenne clinical trials in Europe. And we're initiating registrational cohorts in the DELIVER trial, and we plan to provide an update on the path to registration by the end of the year. So with that, I would like to again thank Little Steps for the opportunity to present to you today. And thank you to the families, investigators, and sites that have made this and so many other advancements in Duchenne possible with your countless sacrifices and dedication. What you all have done over the years and decades is nothing short of extraordinary. And thank you so much on the behalf of Dyne.